All right, everyone, we're going to get started. Thank you, everyone, for being here for this year's Markham Symposium. This symposium is an amazing event where we get to celebrate all of our graduate students and the research that they're doing. We're going to have a keynote speaker from a past student here at Hatfield. We'll have a poster session, and then we'll end the event with some really amazing Ignite talks. But most importantly, just thank you all for being here and being a part of it. We're actually going to kick off the event. I'm going to hand the mic off to Mark Farley. He gave me his official title. I think we should make up a new one. But Director of Business and Operations here at Hatfield. He also had the pleasure of doing some amazing things with today's keynote speaker out at sea. So I thought he'd be a great person to introduce today's speaker. So Mark Farley.
Thank you, Cinnamon. And welcome to the annual Markham Symposium. It's nice to see everybody here. The Markham Awards this year totaled $70,000 and went to 7 students across 4 departments. So $70,000, that was all philanthropic money given to us by donors. So when you hear the loudness of the news these days and the value of science in our kind of federal landscape, think about the louder voice, which are our donors, that actually are in total support of you all. And that's an antidote for a lot of us when things get hard. So I hope that matters to all of you, and those of you who are supported by those funds, good job. So thank you for joining us today. Today's agenda features a keynote. He's standing here awaiting nervously his presentation. And we have a poster session out in the hall set up, and we also have all of our research participants that will be delivering their research in Ignite presentations. So after Will speaks, you're going to hear those students showcasing their research. So I'd like to introduce Will Finney. Will's currently a fisheries oceanographer with Eco-Foci at NOAA Alaska Fisheries in Seattle, where he studies the early life stages of fisheries in Alaskan waters. He has a BS from the University of California, Davis, and an MS in marine science from Moss Landing, and a PhD in integrative biology from Oregon State University. Will spent years here at HMSC as a graduate student, where I had the pleasure of going out to sea with him on one of many of his sampling cruises. In fact, I did a lot of photography work on those cruises, got to know everybody, and I have this for Will. So this sits on my office wall in kind of a gallery of marine science badasses. And Will struck quite a pose, and I want to give that to you so you can have it at home and remember you're always a badass. Although, Will just Will just became a father, and that is the ultimate expression of badassery. So I give you to Will, and enjoy the afternoon, everyone.
Thank you, Mark. Thank you all for having me here today. It's a little surreal being back here telling you about my early career trajectory, because it doesn't feel like I left all that long ago. Can you hear okay?
All the way on.
I believe so.
Everybody can hear? Try
A little
again.
quiet? Better now?
Keep talking.
Okay. So my talk is titled Recruitment Success, an early career study of the factors affecting dispersal and settlement of one HMSC grad. And I'm hoping that this talk will kind of show you at least one student's pathway to getting a job. And hopefully you'll learn some of the lessons, or learn from some of the lessons of my journey, how to make it a little bit easier for you to get to where you want to be in your careers. So a little outline about my talk. We'll talk through my career trajectory, and then we'll talk about some of the things that I found very important for me getting from being a graduate student to successfully having a job. And that's the mentors that I've had along the way, the networks I've created, taking opportunities to meet and work with your heroes. I really want to strike a chord about all the opportunities that are at your disposal while you're at Hatfield and encourage you to take advantage of them because I found them really important on my journey. And then I also want to make sure that you take some time to enjoy yourself while you're in Newport at Hatfield because it's a pretty special place. So when getting into my career trajectory, I'm a self-professed fish nerd, and I like to think of things in terms of fish early life history. And I believe there are some undergraduates in here, and if that's the case, I like to think of being an undergrad as kind of your larval stage where you're trying to figure out how to navigate your early career. You have a little bit less knowledge than some of the people in like graduate school or in postdocs. Is it still quiet?
Really quiet. Let's see. Sorry, everybody. Talk again.
Any better now? Slightly? Maybe? Can it go to 11?
That is really high.
Okay.
Is that better?
Great. Okay, thank you. And so early on when you're an undergraduate, you don't quite have the tools at your disposal yet to really direct your career. But the things that you do early in your career can really set you up for success later on. And then as you progress through graduate school, you become a little bit more competent and have some agency, and so you can make more decisions to help you get to where you want to go. And so we'll talk about how I did that. And my career trajectory started, I grew up on the coast of California. I'm sure, like many of you, I grew up loving being in and on the water, and I wanted to become a marine biologist. I think ever since I knew that it was a thing to want to be something when you grew up, I wanted to be a marine biologist. And so I needed to figure out how to become a marine biologist, and my early love and passion was sharks. That's what I wanted to study growing up. I wanted to be on Shark Week, and I needed to figure out how to have a career doing that. So to accomplish that goal, I went to college at UC Davis, studying biology with an emphasis in marine biology. I chose Davis because it had a marine lab at Bodega Bay, and they had some world-renowned shark scientists, and I figured hopefully that would help me figure out how to become a marine scientist studying sharks. And while I was there, I took some marine science courses as well as ichthyology with Peter Moyle, and I worked in a fish physiology lab with Joe Check. And if you're familiar with this textbook, it's written by Peter Moyle and Joe Check, so I felt like, oh man, I am on the right path. I'm working with these leaders in the field. There's no way I'm not going to end up being a scientist studying sharks. And then in 2008, the financial crisis hit, and Joe lost funding for his lab and couldn't hire undergrads anymore, and I kind of felt adrift. I wasn't really sure how I was going to figure out what it was like to get into graduate school or how to become a marine biologist. And in that time, I was trying to figure out how I was going to complete my marine biology emphasis, and I was encouraged by one of my advisors to take the oceanography program at Bodega Marine Lab, and that's where I met Tessa Hill, and she became my first mentor and really set me on the path that ended up with me being here today. So my first goal was to become a marine biologist studying sharks, and what I want you to see throughout my early career is how my goals shifted and how my plan to achieve these goals changed over time, and how flexibility can be your ally early on when you're trying to figure out where you're going to go. So I had to revise this first goal. I didn't want to just be a marine biologist studying sharks. I also wanted to study oceanography after this course. And then I graduated, and I had no idea how to become a marine biologist studying sharks and oceanography. And so as I was trying to figure that out, I worked at a biotech company for a couple of years, and then applied to graduate school and tried to figure out what that next step would be. And so my goal became I needed to start a marine science graduate program. I wanted to study sharks and oceanography, but I realized that I just needed to get my foot in the door to figure out how to start a career in marine science. So I applied to a couple of PhD and master's programs, and looking back, I am so glad I didn't get into a PhD program at that time. I was not ready. I would have done a horrible job, and I would not be here today. But luckily, I got into a master's program at Moss Landing Marine Labs. There's a couple of Moss Landing alum in the audience here today, which is great to reconnect with. I did not get into the shark lab, but I did get into the ichthyology lab with Scott Hamilton. And that proved to be very fortuitous for me. And so while I was working with Scott, we were trying to figure out what my master's project would be. And during that time, I remembered Tessa Hill in the oceanography course at Davis talking about ocean acidification and how her lab was working on the effects of ocean acidification on marine organisms, mostly shelled organisms. But some research came out looking at the effects of ocean acidification on fish. And I was like, oh, this is super cool. There was these crazy studies showing how rearing fish in acidified water affected their behavior and physiology. And Scott and I both thought this would be a really cool thing to study. Neither of us had expertise in ocean acidification, and we needed to figure out a study species as well, because we obviously couldn't work on coral reef fish in California. But Scott had expertise in rockfish, and they ended up being a great species to study. And then we were trying to figure out how do you go about running experiments with ocean acidification. Moss Landing didn't have the capabilities at the time, but we talked with some scientists down the road at Monterey Bay Aquarium Research Institute. And there, Jim Barry, another former Moss Landing grad, was doing some work on benthic marine organisms in the deep sea and looking at how ocean acidification affected them. And he had space in his lab for certain periods of time during the summer that lined up really well with when we were collecting rockfish. And he allowed us to work in his lab to run our experiments. So this became a way for me to study fish and changing oceanography, and that felt like I was back on track. I knew where I wanted to go. I was excited to study this. And so we went out and collected blue rockfish and copper rockfish and ran them through a gamut of behavioral and physiological tests at different ocean acidification treatments. And we expected that copper rockfish might be a little bit more sensitive based on their early life history. They hung out higher up in the water column where there was more photosynthesis happening and lower CO2 in the water. And they recruit to kelp forest canopy where it's the same conditions there. And blue rockfish hang out a little bit deeper in the water where there's less photosynthesis, more CO2 buildup, and they recruit along the benthos and kelp forests where upwelled water brings really low pH water into the system. So they might have been a little bit more resistant. And that's exactly what we found. We found these species-specific responses to ocean acidification and their behavior, their physiology, and their genetics. And at the end of that, I realized, we only saw those effects at the really extreme conditions. And it didn't seem like ocean acidification was necessarily going to have the strongest impact on rockfish moving forward. But I really enjoyed my time working with rockfish, and I kind of wanted to figure out where I would go next. And while I was at Moss Landing, I got introduced to working for NOAA by being recommended to take a summer to work on their groundfish surveys. And so I ended up coming up to Newport in 2012, I think, was my introduction to Newport. And I learned how to be a volunteer on their groundfish surveys. I went out a couple weeks later out of Coos Bay and sampled groundfish all the way down to San Francisco Bay. It was a really cool experience. I remember being really impressed with the work that the scientists were doing, kind of enamored with the idea of working for NOAA, and that sort of stuck in the back of my mind throughout my time at Moss Landing. And then Moss Landing was a really special place because it empowered its students to become independent field scientists. They allowed you to check out tanks, check out boats and a trailer, and drive anywhere along the Monterey Peninsula and go diving. And so I had a ton of fun with my advisor and my lab mates going out there and collecting juvenile rockfish. And during that time, I really kind of fell in love with kelp forest ecology and wanted to know more about what was going on in the early life history of rockfish. So that kind of led me to reevaluating my goals. So my original goal was to complete a marine science graduate program so I could be a marine biologist studying juvenile rockfish and ocean acidification. And as I accomplished that, I found that I was more interested in how oceanographic conditions affected the early life history of rockfish, especially their growth and survival. And I wanted to get a job at NOAA's Southwest Fisheries Science Center studying rockfish. So that changed my goal going forward. I talked with Scott and I was asking him, how does one get a job doing that? And he said, well, you're going to need a PhD. I'm like, oh, great, there's another half decade of my life. It also became really apparent how important choosing the next advisor I worked with would be to my career success. I was talking with Scott about who would be good people to work with. And he mentioned Mark Carr at UC Santa Cruz. And he also mentioned that there were some people who did some really cool coral reef work out of University of Miami. They're actually moving to the Oregon coast and might be looking for a student to start doing some of the similar work they were doing in Miami out on the West Coast. And that was Bob and Sue. So I contacted Sue, and she can correct me later or now if she wants. But I think the reason that Sue took me on as a graduate student was because I had thought through how I wanted to go about trying to use some of the techniques that Sue's lab was famous for, tracking cohorts of fish across their early life history and figuring out what traits were influenced by oceanographic conditions and affected their survival to settlement. I was hoping to do that with rockfish, and I think Sue recognized that I had some experience collecting rockfish, and maybe I tricked her into thinking that I'd do a good job of this at Oregon State. And then I asked her, you know, I think my career goals are to work for NOAA studying juvenile rockfish and recruitment drivers, and that was something Sue was really supportive of as well. I got a little bit ahead of myself. Anyway, so Sue took me on, I think, and was excited about the reasons why I had kind of professed her what I wanted to do with my career. And so going forward to catch these small fish in Oregon, I realized it wasn't going to be quite as easy as in Moss Landing, and I was going to need a bigger boat and some friends. And that's where Sue really helped connect me with some collaborators at both NOAA and at Oregon Department of Fish and Wildlife that really helped me build this program that I was hoping to do for my PhD. And one of the people she introduced me to was Rick Broder. I don't know, I think everybody here probably knows who Rick is, but in case you don't, Rick is a former fisheries oceanographer at the Northwest Fisheries Science Center. He ran the rockfish pre-recruitment surveys out of Newport here for years. He's incredibly generous in having people come out on his surveys and work with him to get samples. So he had me come out, and I was going to collect pelagic juvenile rockfish as part of my PhD with him. And then Sue introduced me to one of her former students, Kirsten Grorud-Colvert, who was actually in the integrated biology department already. And Kirsten does a lot of work with marine protected areas and had developed this sampling program to monitor near-shore settlement of juvenile fish inside and outside of marine reserves. So I worked with Kirsten and her collaborators at ODFW and the Oregon Coast Aquarium to collect rockfish as they were transitioning from their offshore life stage to their near-shore life stage. And then my skill sets for Moss Landing of doing subtidal collections of rockfish came into play when we were trying to collect them on reefs. And we had some great lab mates and a lab manager, Jamie Ivory, Megan Wilson, among others, who dove with me to collect juvenile rockfish once they'd settled to reefs. So I had an idea of how I was going to go about collecting rockfish across their early life history to mimic the work that Sue had done in coral reef ecosystems. Now I just had to figure out how I was going to analyze data from them. And that's where another one of my committee members really shined. So I got the pleasure of working with Jessica Miller on this. And Jessica is also an otolith expert, but she has particular expertise with juvenile black rockfish from her PhD. So she ended up being my guru for analyzing juvenile black rockfish otoliths, and she also had a lot of suggestions about how to go about collecting them on the Oregon coast. So I really valued working with her. And I got a really cool set of opportunities to do work with rockfish otoliths. Sue, I remember, had presented us with a paper from Akinori Takasuka out of Japan, where he'd looked at selective mortality of bay anchovy by a group of predators and showed that there was selective mortality on anchovy based on their early growth rates, and you could track that with otoliths. And Rick and I had talked about doing that a couple of times, and we hadn't quite figured out how to do it. And then one summer, the stars aligned, and his pre-recruit survey teamed up with the Juvenile Salmon Ocean Ecology Survey. And we were able to go out and collect salmon and rockfish at the same time. And then I pulled rockfish out of the stomachs of the salmon and compared their early life history traits And what we found really matched up with what Takasuka et al. Had found, and that the consumed rockfish in the stomach of salmons were younger, smaller, and slower growing at the time of capture. And this was a really exciting finding, at least for me, because it met the expectations that people had previously shown. And I was able to get a project out of one cruise for my dissertation. And this paper actually was kind of the springboard that got me my postdoc, and we'll talk more about that later. And next, looking at kind of the culmination of my work, studying the effects of oceanographic variability on rockfish on the Oregon coast. What we found is that when conditions were warm, it really increased the growth rates of rockfish. And you can see here, in warmer years, which are the warmer colors, growth rates rapidly increased for rockfish compared to in the cooler years. And this led to them transitioning from their larval stage to their juvenile stage much earlier. But I was a little surprised to find that we couldn't figure out how oceanographic conditions affected their settlement. And it turns out that it was mediated by growth rate. So growth rate kind of combined all the factors that the oceanographic variability had on rockfish and translated that into their recruitment success. So on this figure here, we have growth rate on the x-axis and settlement on the y-axis. And in years when it was colder, that led to slower growth, and that meant fish were more likely to be consumed by predators. In years when it was warmer and they had really fast growth, only the few fish that were able to find enough food to sustain their metabolisms that were elevated by temperatures were able to survive. And it was this sweet spot where conditions supported fast growth, but fish could find enough food to support it, that we saw really high recruitment. And I remember when I plotted this up, I almost started crying because I knew that I had kind of achieved my goal of trying to figure out how oceanographic conditions affected early growth and survival of rockfish, and that Sue was likely going to let me finish my PhD and move on. So this is a really important figure for me, and I hope you all find similar ones in your work. So now my goal was to complete my PhD studying the oceanographic drivers of rockfish early growth and survival, and the next goal was, all right, now it's time to get a job. And there weren't a whole lot of jobs out there at the time that I finished my PhD. There was this weird thing going on that was called COVID, and not a lot of people were hiring. So my goal was, okay, well maybe I can at least find a postdoc. And the way that I found a postdoc is, I'm surprised that it worked out, but I'll tell you the things that I did that I think helped me get it. So while I was working on that project looking at salmon predation on juvenile rockfish, I was working with Michael Banks and Dave Jacobson to genetically identify the rockfish that had been consumed by salmon. Sue won't let me show the photos of them, but basically if you had like snot that kind of looked like fish, that's what we were trying to identify the species. And so we needed some genetic techniques to do that. And the person who had developed them was Andrew Thompson at the Southwest Fisheries Science Center. And so we reached out to him to kind of confirm some of the methods that he had done for his DNA sequencing, and he was so excited to hear what we were doing. He was like, Will, this sounds super cool. Send me the paper when it comes out. I can't wait to hear about it. He sent us the sequences that he used to identify fish to species. I was like, man, that guy is super excited about rockfish. I think I'd do really well working with him. And so at one point, someone shared with me NRC postdoc list, and I couldn't believe it, but on there was a larval rockfish otolith postdoc position with Andrew Thompson. I was like, there's no way this is real. But I reached out to him, and it turned out that was an outdated listing. He didn't have funding for it anymore because he'd already had a postdoc working on it. But they were about to finish, and he's like, send me some of your ideas on what you might do with this, and send me your paper, and we'll see if we can figure something out. And so I wrote up a brief proposal of what I would be interested in doing with larval rockfish otolith work, because his postdoc was moving on before they'd finished analyzing the data, so there was potential for me to work on it, but not funding. And when I sent him my paper about the salmon predation work, he's like, oh my gosh, this is so cool. Okay, yes, I want to hire you. We're not going to necessarily get to work on rockfish, but we will get to work on anchovy. Are you in? I was like, sure, Andrew. That sounds great to me. I needed a job, and I knew I wanted to work with NOAA, so I was happy to change my goal from working on rockfish to completing a postdoc looking at the effects of variability in anchovy distribution on higher trophic levels. And I had the side goal of helping him complete that project, looking at environmental drivers of larval rockfish growth and survival. So I was at the Southwest Fisheries Science Center. I was working for NOAA. I was getting paid by them to work for NOAA. And it felt like I was kind of in a really good spot. So now I just had to figure out how to get a permanent position studying rockfish at the Southwest Fisheries Science Center. And I figured the best way to do that was to do a really good job on my postdoc. And hopefully the people there would recognize the work that I had done, and when a job opened up, I would be in a good spot to get it. So that anchovy project, I'll tell you a little bit about it and how I think I used it to gain skill sets. So it was this really collaborative project where it spanned fisheries economists, fisheries oceanographers, physical oceanographers. I remember I had never heard about species distribution modeling before starting this postdoc, and I felt totally out of my zone. It wasn't rockfish, and it was not otolith work, so what was I going to do? But it had this really kind of crazy tempo to it where we would meet every week, and you'd have to give updates, and there was just data sets, so there was no field work. It was just crunching numbers. And I was like, okay, I want to work for NOAA. I need to figure this out. And so I started just kind of cranking things out, learning about species distribution modeling. And then one of the things that they wanted someone to do was to validate that these models actually translated to reality. And so I was like, well, do we have any data sets looking at things that eat anchovy? Can we align them with the model output from this? I was like, well, that sounds like a great project. Go work on that. It was kind of terrifying to go out of my comfort zone, look at things like seabirds and marine mammals, which I'd never studied before, and present updates on this to 12 to 15 scientists who were almost as intimidatingly intelligent as Sue, but there was 12 of them. It was terrifying. And one thing I will recommend to all of you is that if you can possibly avoid it, never have a draft of your manuscript live edited by 12 people while you're watching on Google Docs. It might be the most terrifying thing I've ever done besides being a parent. So avoid that at all costs. But this ended up being a really cool project. So I took the model output from Barb Mulling's species distribution model, and I found that there were data sets of seabird foraging on the Farallon Islands and sea lion foraging in the Channel Islands. So we tailored this model output to the foraging ranges of those species, and then looked at how well it captured variability in their diet, their distribution, and their reproduction. And what we found is that it did an excellent job predicting variability in seabird diets, especially in how much anchovy they were consuming. And then when we looked at the variability in their distribution, we found that most species followed these areas of really high anchovy abundance. And that makes sense because they were eating so many of them. And then we found that it explained variability in Brandt's cormorant and California sea lion reproductive success. And that definitely made sense for the California sea lions because in years when there were more fatty fish like sardines or anchovy, their pups were in great condition and more likely to survive. But when they were eating things like rockfish or squid, they tended to be lower energy content, and it wasn't as great for the survival of their pups. And then I got to work with that larval rockfish otolith data set, and it was a huge sigh of relief. I wasn't having those crazy meetings. I was back in my comfort zone, and I thought, okay, this is my opportunity to really wow the people at the Southwest Fisheries Science Center. So I was going to dive full in and try a whole bunch of new things with it. So I worked with the former postdoc who had generated the otolith data set. We brought in some oceanographers who were some of my heroes, and we'll talk a little bit about that later too. And it was really rewarding to squeeze this into my postdoc. So what we did was we ended up looking at how oceanographic conditions that female rockfish were experiencing during gestation affected the quality of the larvae they produced and their early life history traits. And we found that when there was a less, influence of Pacific subarctic upper water in the California current, female rockfish produced smaller larvae that grew slower and were less likely to survive. And this happened across like 8 species that we tested. And in contrast, when there were higher concentrations of Pacific subarctic upper water, that led to what we thought was better feeding conditions for the females, led to larger larvae at extrusion that grew faster and were more likely to survive. And this was my favorite paper that I've gotten to work on. And I'll talk about that in the Meet Your Hero section. But at that point, I felt like in 2 years, I'd done 2 really cool projects during my postdoc. I felt like I was probably ready to get a job at the Southwest Fisheries Science Center. So that was my goal at that point, was to wrap up these papers, get them published, and then try and get a job. And they weren't hiring. And there was no real indication that they were going to be hiring anytime soon. I was trying to figure out what I was going to do next because my postdoc funding was running out. And then I got a call from one of my lab mates from Sue's lab, who was working at the Alaska Fisheries Science Center. She said, "Hey, Will, we're hiring somebody to look at environmental drivers of early life history traits of pollock in Alaska." I was like, if you replace pollock with rockfish in Alaska with Southern California, I think I have a chance at this job. I just have to be a little bit flexible on the things that I'm thinking about, and maybe it might make sense. I still didn't have those papers out yet. I'd only had that rockfish predation paper, or the salmon predation on rockfish paper out. So I wasn't super confident that it was going to work. And I was telling Keely about this, and she was like, "Will, these jobs don't come around very often. You have to apply. You're almost perfectly qualified for this, and you definitely won't get the job if you don't apply." I was like, okay. It's a pretty good argument, Keely. It's great to have good lab mates. And so I applied, and I took some advice from both Sue and from Andrew in my approach to the interview process, because I got to the interview stage. And Sue is one of the most prepared people I know. She made me prepare for one of my talks at a larval fish conference like 1,000 times. So I knew that I was going to do what Sue would do in preparing for this job interview. And then Andrew told me the reason he got his job at the Southwest Fisheries Science Center was because he absolutely knocked the socks off his interview. They were actually trying to hire somebody else who already worked for that group, but they didn't take the interview very seriously, and Andrew created this whole research program idea for what he was going to do for the cow coffee program as an outsider coming in. And when he gave his presentation, they were amazed, and they're like, "We have to hire Andrew." Even though we thought we were hiring this other person, clearly Andrew's the right person for this job. And so I treated the 2 weeks in between when I found out I was going to get an interview and that interview as like a mini orals exam prep session. I read all the papers I could on the physical oceanography of the Gulf of Alaska and the Bering Sea. I looked at all the work that group was doing on the early life history of pollock, both inside and outside of the Gulf of Alaska and the Bering Sea, and figured out, okay, they're doing a little bit of otolith work. They don't really have a whole lot of otolith growth work going on. They have like a 30-year time series. I think I could make a pitch that if we use that whole otolith time series with some of the stuff that I've had experience with species distribution modeling, with some of my rockfish otolith work, I think I can come up with an idea for this program. And so I got through the first round of interviews, and they said, okay, the next one's going to be in 2 weeks. And I think I was told like to come up with like a vision for your research program for the group. And so I thought about what I wanted to do in the first year, the first 5 years, and the entire career that I'd be at that program, and pitch them different projects at each of those steps. And I think that's what got me the job in the end, was that they saw how well prepared I was, what vision I had for how I'd use their data sets, and that I was clearly thinking about both the short and long term of my career with that group. And they hired me, which is still a little shocking to me to this day, but I will not argue with it. So now my goal is I found a job studying the effects of environmental drivers of recruitment variability on walleye pollock in the Gulf of Alaska and the Bering Sea. I work at the Alaska Fisheries Science Center in Seattle. I do field work in Alaska twice a year on the spring ichneoplankton surveys, and then I lead the Gulf of Alaska juvenile pollock survey. And I'm in the process of building an otolith time series, both spawn timing and growth rate. And I plan to be doing this for as long as my wife will let me do it. And so that's my career trajectory. And now we'll move on to the importance of mentors to getting me to where I am today. I think I've given a little hints of it along the way, but we'll get into the specifics of that. And among many things, mentors' job is to teach you how to be a scientist, to instill confidence in you as a scientist, to help you develop skills, to get the best out of you, and to encourage you to progress in your scientific career. And it's really fun to look back at my career and think about how my relationship with my mentors changed as I advanced through my career. When I was at UC Davis studying with Tessa Hill, it was really top down because I didn't know anything, and Tessa knew almost everything. And so she was the one that got me interested in oceanography because, like, how can you study marine organisms without understanding the world that they live in? And that kind of like built as I got more and more advanced in my career. It became more collaborative. I now had agency, I had skill sets, I had ideas, and so I would work with my mentors on achieving them. And now it's almost like I have all this stuff at my disposal, and I'm just looking to my mentors to support me in executing them. And I want you to remember that you choose your mentors as much as they choose you, and you also want to get the best out of them. And I think some advice I could give for how to go about finding mentors is to think about what you're interested in that they do that you would like to do, and propose to them, you know, like for example with Sue, I wanted to study rockfish early life history. I wanted to track cohorts of rockfish from their offshore life stage to their nearshore life stage. I knew Sue had expertise in this. She did otolith work on tropical reef fish with this. How was I going to pitch to her, this is my research program that I would like to do. This is where you're really good at these kinds of things. Can you help me figure out how to do this for a PhD? I think that's how I got Sue to be my mentor. Sue, feel free to argue with me about that later. And so I think that's what I want you to think about as you're moving through your career and trying to pick mentors, is pick people who have research interests that you share, but have the capacity to help you advance yourself with those research projects that you want to do, if that makes sense. And then we'll talk a little bit about the individual mentors that I have and what I wanted to take away from them as I advanced in my career. So with Tessa, I wanted to become an oceanographer because of her. And I think the main takeaway I wanted to have was that she believed in me and had confidence in me in being a marine scientist. And so I tried to incorporate Tessa's confidence in me moving forward. With Scott, he was my master's advisor, if you might recall from a couple minutes ago. I came into his lab at a really cool time. He had just started. I was one of his first 2 students, and so he had a lot of time to invest in me as a student. And what he really helped me with was building capacity as a field scientist. And what he did was he trained me so that I could then train his future students coming in, because he was going to have less and less time to do that. So that was a really cool thing where I learned from Scott and then took what I learned from him and taught others. And that was a way of building me up as a scientist. He also began honing my writing and scientific mind and encouraged and guided me to finding the next step of my career path. So from Scott, I tried to take his excitement for field work and his work ethic. Going back to Scott real quick, I have no idea what I said to him to get him to take me as a student, by the way. I think that was just luck. With Sue, I had a much better plan for it, and I think that's just from experience. So sometimes you just need a little bit of luck. And then moving forward, once you kind of figure things out, that's where you can be a little bit more thoughtful and prepared to take advantage of finding mentors and moving through your career. So yeah, Sue, as my PhD advisor, you all know she's a world leader in fish and wildlife history ecology. She's incredibly supportive, funding for me and my work, and provided some really unique opportunities for me through lab cruises. That's where I got to work with Mark Farley a bunch and be a model for him. She also provided some cool collaborations through NOAA and ODFW and Oregon Coast Aquarium, and through that, I was actually able to complete my research at OSU. She also pushed me to know my field inside and out, and that served me so well when it came to oral exams, when it came to applying to a postdoc, and ultimately getting my job. So thank you for that, Sue. And between all of my advisors, Sue is the best editor I've worked with by far. Thanks for looking out for me, Sue. Sue is the best editor I've ever worked with. Yeah, that's better. And she prepared me for my dream job of working for NOAA. And so what I tried to incorporate from Sue was her precision, her preparation, and her attention to detail, at least to the best of my ability. Next, I want to talk about Rick Broder and how I got to work with him. There's at least a really direct line between my master's program and Rick and working with Sue. I'll talk a little bit about that later, but basically, you all know that Rick is a famous fisheries oceanographer. And while working with him, I got an idea of what it looked like to be a federal scientist doing fisheries oceanography work, and I could actually see myself having the same job that Rick had. So that kind of affirmed my interest in being a NOAA scientist. Rick is also incredibly collaborative and generous, and so what I tried to incorporate from Rick is his humility, and I endeavored to be as generous as he is. And then for Andrew, he's the lead Cal Coffey early life history ecology researcher at the Southwest Fisheries Science Center. He is by far the most enthusiastic scientist and person I've ever met. We're on a paper together where he got reviews back, and he was criticized for using adjectives like meteoric, famously. I can't even remember what some of them were. I was like, gosh, I need to write these down, and I would love for reviewers to critique me for being as excited about science as Andrew is. He really modeled collaborative research with an amazing data set, provided some really cool opportunities for me to broaden my skill set, taught me how to navigate the federal bureaucracy, which I am still struggling to learn today. And what I tried to incorporate is Andrew's collaborative nature and his enthusiasm for science. And finally, my current mentors are Libby Lagerwell and Julie Keister at NOAA AFSE. I'll always be indebted to them because they hired me, and I'm basically doing my, if you squint really hard, I'm doing the dream job that I have when I started my PhD with Sue, if you just change a couple words around. They give me almost complete research freedom. They support my professional development by recommending me to working groups and international organizations. They also encourage me and expect me to find new ways to use our data sets. It feels really empowering to have people trust you to go out and do new things. And so I'm trying to incorporate the breadth of the research scope in my work. I've worked locally in California, I've worked kind of regionally in Oregon and at the Southwest Fisheries Science Center. And now I'm trying to think a little bit more broadly because Pollock kind of spanned all the way across from Alaska into Japan. And so I'm trying to build up international collaborations and do some work kind of spanning the whole North Pacific. Next, we'll talk about the importance of networks. I think you've already heard a couple of points throughout my career where my networks really benefited me. And I'm going to help you figure out how to build networks and the value of them. So they're inherent in graduate school. If you have an advisor, you already have the start of a network, especially if they have a lab and lab mates, you're already starting to see your network build. And as you meet more people and go to conferences, it just kind of builds and builds and builds. And networks are great because they can expose you to new ideas and techniques in your field. They can also alert you to funding or job opportunities. And science is increasingly collaborative these days. So building a network can increase the amount of research you do and the scope of the research you're able to accomplish. So I'll show you kind of how my network with Sue was built. So it starts off with Sue, and then it flows into my committee members. And then from there, you can see how all of a sudden you're exposed to really cool science and opportunities. So through Sue and meeting Rick and going out on his recruitment surveys, I got to work with a bunch of NOAA scientists. I got exposed to traditional fisheries oceanography work. I got to see what it was like to be in that field, and I could picture myself in that job. Also, it became really important. So on the bottom here are some people that work at the Southwest Fisheries Science Center. And so when Rick would come back from a survey, I would take all the juvenile rockfish from the survey, drive them down to Santa Cruz, and I would go to Keith Sakuma's lab, and he and I would sit together and identify all the rockfish to species. So by working with him, I learned how to identify pelagic juvenile rockfish. And then from Tom Leidig, if you're aware of him, he was an expert in nearshore rockfish ecology. And without him, I wouldn't have been able to identify or distinguish between black and yellowtail rockfish from the Smurfs. And without that, I couldn't have done my project. And then through Jessica Miller, that's how I actually first heard about ecofoci, which is where I work now. So Jessica introduced me to Ben Laurel, one of her collaborators. There's also Tom Hurst and one of Jessica's students, Hillary Thalman, who have all kind of worked together on Alaska species. And through them, I got to learn about ecofoci. And then through Jessica in particular, I got to figure out how to be an expert on juvenile black rockfish otoliths. And then I already talked about how Kirsten introduced me to the joint program with ODFW and Oregon Coast Aquarium. That's the SMURF program. And without them, I wouldn't have been able to go out and collect settlement stage juvenile rockfish. It's some of the most fun field work I was able to do while I was here. And if you ever have the time and interest, I'm sure they would love to have volunteers out there. You can find Ryan Field. Ryan, raise your hand. Talk to Ryan if you want to get out there and go do some smurfing. It's a blast. And finally, through Sue, there's the whole lab community. And I don't think I would have survived my PhD without my lab mates. For one, they're really helpful and collaborative, getting out in the field and helping you sample. Two, we kind of created our own grad student-led lab meetings in preparation for our lab meetings with Sue. I don't know if Sue knows about that, but Sue was, it took a lot of preparation to have successful meetings with Sue, and I think we all found that we did a better job of that when we worked together and workshop things before presenting them to Sue. So I love all of my lab mates. I think several of them are coming into town this weekend. We're going to hang out and enjoy time in Newport together. So having a great lab and great lab mates is really critical to your success. It's also critical to finding jobs, apparently. Thank you again, Keely. So yeah, here's just the snapshot of a network that I built through Sue. And these kind of stack on themselves as you go throughout your career. So it was kind of cool to look back at being at the Alaska Fisheries Science Center and then looking down at all the levels of networks that I built that helped me get there and how they interact. And so I'm going to show you kind of briefly some kind of crazy connections that happen and help me get to where I am today. So while I was at Moss Landing, I took a class called Scientific Methods that was led by Jim Harvey. He was the former director and former Marine Mammal Lab PI. In this class was a mixture of statistics, philosophy, and like a mini oral exam. And so I got to know Jim through this, and he did his PhD at Oregon State. And while I was telling him about my plans to start a PhD at Oregon State, he was like, oh, I'm going to have my old friend come down and give a seminar. Why don't you give him a ride back to the airport and you guys can talk about things? He's like, okay, that sounds cool. Who is he? He's like, oh, his name's Rick Broder. And so Rick came and gave a seminar, and I drove him back to the airport. And in that like hour-and-a-half car ride, I talked with Rick about wanting to work with Sue and my interest in rockfish and wanting to work for NOAA. And he was like, hey, I have a survey in May. Do you want to go on it? You can take samples. And he ended up being like a critical component of my PhD, and it's all thanks to this one car ride that Jim Harvey voluntold me to do. So if it wasn't for that, I think Rick probably would have ended up on my committee anyway, but I'm not sure I would have gone out on that first survey without it. And so that ended up being a really fun connection, and it was really important to my career. Next, and this is where you find out how small the marine science community is. So my master's advisor, Scott Hamilton, actually went to UC Santa Barbara and was a grad student with Andrew Thompson. In fact, Andrew taught Scott how to surf. And so when I was asking Scott for a letter of recommendation to apply to the postdoc with Andrew, Scott called me up and was like, Will, Andrew's one of the nicest people I know. He loves to surf, he loves rockfish. You guys are gonna get along famously. And I think he told Andrew, like, you should take Will. And I think that was very helpful, having Andrew know someone that he trusted, tell him that I'd be a good fit for his lab. And then there's all these like interstitial connections. So Scott now works closely with people at the Southwest Fisheries Science Center because he has an ocean acidification and hypoxia lab that he built based on funding he got from the ocean acidification work that he and I did together. So he now works with the Southwest Fisheries Science Center, especially with John Field, and then John and Rick and Toby all work together with Andrew in putting together the State of the California Current report. A quick shout out to that report for any of you doing oceanographic work along the US West Coast. Definitely check out the State of the California Current because it compiles the physical oceanography of the system in any given year and builds up to the biological oceanography, zooplankton productivity, all the way up to fish, to top predators, including birds and mammals. So anything you want to know about changes in abundance or patterns within the California Current, check out that report. So they all work on that with Andrew. And then through Sue and Jessica Miller and Ben Laurel, I was introduced to people who work at EcoFoci. They work closely with Lauren Rogers, who's a quantitative ecologist in our group. And I now work closely with her on trying to figure out how spawn timing varies with pollock in the Gulf of Alaska and the Bering Sea. Andrew also worked with Jens Nielsen, who was a former postdoc in our group and is now a cooperative institute member. And so Jens and I talk a lot about recruitment variability. And then, of course, without Sue having Keely in her lab, who got a job at EcoFoci before me and called me and told me I had to apply for that job, I wouldn't have the job. So this is how a network can really benefit you and help you get through your career. And it gets a lot more complicated than what I showed you. And so you start building these as an undergraduate student. And as you progress through your career, you'll find that these interactions kind of mix and match and build their way up to the point where you have this huge network. And through that network, you're going to find really cool opportunities. And if I have time, I'm going to talk through how important it was for me to meet my academic heroes and to work with them. So in 2019, this paper came out by Schroeder et al. That kind of changed my understanding of how oceanographic variability affected recruitment of rockfish in the California Current. I remember we did this as a lab group, Sue, where we talked about how Pacific subarctic upper water and its influence in the California Current affected recruitment processes. And this was foundational for my understanding of my PhD. And I remember reaching out to them to see if I could use their indicator in my PhD work. And unfortunately, because I had such a short time series, I don't think I was able to find any relationship with PSUW and rockfish recruitment. But during my postdoc, when I was looking at larval rockfish otoliths, and I had a longer time series, I knew that this is where I could leverage that project and those scientists to help me figure out what was going on. And this ended up being really cool for all of us. So when I reached out to them, they're like, oh, cool. Someone's actually using our indicator in a way that we hadn't quite thought of. And they found it was like, that's how science is supposed to work. You put something out there, someone reads it, they build upon it, or they pull you in to like figure out how it could be applied in new ways. And then for me, it was, I got to meet my heroes and work with them, and it felt really cool to see the work that I was interested in being influenced by their work, and that was really fun. And finally, I think I already talked about this, but getting to work with Andrew was a moment that I got to meet one of my heroes, because he was so excited about the work I was doing with salmon predation on rockfish. And so getting to work with him was a dream come true. And now I want to extol the virtues of Hatfield to you all. For those of you who are in the RU program, good job. You're way further along in your early careers than I was at your stage. For those of you who are graduate students who are thinking about having an RU student, I highly recommend you do it. Especially if you want to be a mentor and be on people's committees, it's a great opportunity to start to learn how to mentor students. And you get someone to help you do your work. It's a really cool collaboration. I know you're all aware of this, but there's many different labs and agencies that work in and around Hatfield. And if you're trying to figure out what you want to do in your career, I highly recommend you go talk to people in different labs or who are doing different jobs than what you're currently doing, so you can get a sense for what the landscape of your career could look like. And you might find things that you're more interested in than you realized. And then there's tons of opportunities for teaching and outreach. There's World Ocean Day, there's Marine Science Day, there's Science on Tap, and you can volunteer at the aquarium. And those are all different ways to interact with the community and help people understand science that we're doing here. Yeah, as I said, if you're not sure that academia is exactly where you want to see yourself in your job career, go look at people who work at USDA or EPA or ODFW or the aquarium or maybe NOAA, and ask people. I think if you come to people and say, hey, I'm really interested in what you do, can you tell me a little bit more about your job, how you got there? And I guarantee you people will be excited to talk to you and help you figure out how to find a career that's similar to theirs. And finally, I want all of you to make sure that you have as much fun as you possibly can when you're not working while you're in Newport. I found I've been away for like 5.5 years, and I was so excited for the opportunity to come back, because some of the best years of my life were spent here. This is a picture of me and my lab mates going spearfishing just off the third finger on the jetty there. And some of my favorite times were us spearfishing and making fish tacos as a lab. And so I hope that you all find similar things to do in Newport that keep you grounded while you're getting ground down by your master's or PhD. And finally, I wanted to show a couple of observations of former OSU and Hatfield graduates in the wild. So last summer, I went out on a Eastern Bering Sea groundfish survey, and I was on the same crew as Erin Fedewa, who's one of Jessica Miller's former students. It was really fun getting to work with her. She's in the Kodiak crab group for the Alaska Fisheries Science Center. And so is Shannon Hennessy, who's one of Mark Novak's students, and she and I were in the same cohort in integrative biology. So she was on the companion boat while we were out there. And of course, Keely Axer is one of Sue's students, and we now have offices right next to each other. So also with your networks, don't burn any bridges, because you never know who you might end up sitting next to in your new job. And with that, hopefully I've helped you kind of understand how one person navigated their career. And if you have any questions about what I've done that might be helpful for you, please don't hesitate to ask. I should be around for the poster session and the banquet as well. So if there's something you don't want to ask in front of an audience, please feel free to come up and talk to me one-on-one. Thank you very much.
All right, everybody. We're going to get started. I think I've lost a couple folks to the snacks out there, but we'll try to get them in here as soon as we can. Thank you so much for being here and being back. We're going to go into our lightning talks. These lightning talks, if you haven't been in a lightning talk session before, happen really fast. There's no questions in between, and each of our presenters, their slides are set up to advance on their own. So they have to figure out how to keep the cadence going and keep up with it. So it is really an interesting and fun way to get a really fast overview of the research that's coming through. And so the students that are going to present today, we have a mix of folks that are here in person and a couple of folks that have given us a recording of their talks because they either have graduated or they're out in the field today. So you'll see a couple different types of formats, but these are folks that have won awards either this year or last year. No, I'll start again. This last year, so they are just finishing their award, or they won a Markham Award, which is a 2-year award, so they've been working on their projects for the last 2 years. So you're seeing kind of the end point of their projects. There will be no questions in between, and our presenters are all up here in front. They're going to hand the mic forward. Just a reminder, since we had a little trouble with the mic before, for my presenters, if you can put the mic like right at the center, that will help us make sure we can hear you. I also just want to remind the presenters to stay in the blue box if you can, so that those folks online can hear us as well. Anybody joining us online, thank you so much for being here. If you have any technical issues like you cannot hear us, please, please, please let us know in the chat box, and we'll continue to adjust those mics so that you can interact with us as well. So we're going to get rolling here with our very first presenter. So we have Sasha Langley, who is working, I need glasses because I'm old, working with James Kulikowski and Bernarda Kalla. He won a 2005 Marianne Boza Award and a Holt Award this year, and is working on almost getting ready to complete his master's. So, where's Adrian? Adrian is not here, so there might be a little photo shoot that needs to happen, but it might have to happen after if we can't get it going now. All right, go ahead. We'll just get us rolling. All right, round of applause for our first presenter.
Can you all hear me? Can online hear me?
All good?
Wait 1 second.
Okay.
Wait 1 second. Yeah. Okay. Let's go. Good afternoon. My name is Alexander Langley, and I'll be discussing how harmful algal toxins may be impacting the Pacific oysters on your dinner plate. So, without further ado, let's dive right in. The quickest explanation is to blame it all on climate change. That's it. So, thanks for listening. No, I'm kidding. There's more to it than just that. Let me explain. The ocean absorbs much of the carbon dioxide that we release into the atmosphere. As CO2 levels rise, oceans warm, become more acidic, and marine diseases become more prevalent. While these conditions stress most marine species, harmful algae absolutely love them. This has amplified the frequency of harmful algal blooms, HABs, along coastal areas. Many HAB species along the Pacific Northwest coast produce toxins that harm wildlife, pets, and even people through different types of poisoning. That's a major concern for shellfish agriculture. Shellfish provide food and income for coastal communities throughout the Pacific Northwest and around the world. Shellfish are also incredible filter feeders. Oysters are a great example. Here in this video, we can see how they maintain water quality by removing the phytoplankton and other suspended particles from the water. When oysters group together, they form reef structures that provide habitats for a variety of marine species and help provide shorelines protection against coastal erosion. But it's not all sunshine and rainbows for oysters. Here in the Pacific Northwest, oysters regularly experience mass mortality events, especially during the summertime when temperatures are much warmer. Scientists have linked these mortality events to warming seawater, ocean acidification, and disease. These stresses impact oyster physiology and increase infections from pathogens like OSHV1 and Vibrio. But what about harmful algal blooms? Their role in these mortality events is less much understood. Evidence from the Pacific Northwest suggests that HABs may contribute to oyster summer mortality. HAB toxins can affect shellfish in many ways, altering their feeding behavior, morphology, growth, reproduction, and overall survival at both the juvenile and adult stages. Toxins produced by Alexandrian species, a common hab species here in the Pacific Northwest, can affect embryo and larval growth, feeding, settlement, and survival in Pacific oysters. Unfortunately, when these toxins accumulate in shellfish tissues, they become a health risk to wildlife and people. That's why hab outbreaks often lead to shellfish harvest closures. So, do I have the end-all solution to this problem? Absolutely not. But I can help fill in one piece of the puzzle. I exposed juvenile oysters to toxic and non-toxic Alexandrian species to compare their responses. Because oysters are picky eaters, I measured their clearance rate, the amount of algae removed from the water over time, to determine how different algal treatments influence oyster feeding behavior. I then measured toxin levels and extracted RNA from those oysters to investigate how feeding behavior affects toxin accumulation and how exposure to those toxins alter genome-wide gene expression. I'm still working through my data, however, one thing stood out in the clearance results. Oysters cleared the toxic Alexandrium at a higher rate than the non-toxic and dietary species. As to how toxins affect oysters, well, we sequenced RNA from 45 oysters and generated 4.1 billion reads. These reads should help us identify which genes are expressed at higher or lower levels during toxin exposure. And with that, thank you to my advisors and committee, the labs and lab members involved in all of this work, and the 2 HMSC scholarships that provided me the opportunity to present here today. Thank you for listening.
Perfect. All right. This is going to go fast. So our next presenter has recently graduated and is unable to join us today. Madison Vargas, who worked with Cheryl Barnes, received 3 different awards last year: the Cribbins, the Holt, and the ODF&W award that helped her finish her research and complete to graduation. And so we have a video from her. Maybe.
My name is Madison Vargas, and I'm going to share my work on my master's thesis for this year's Markham Symposium. My master's thesis focuses on spatial variation and life history traits of black rockfish in the California Current and Gulf of Alaska. So the fundamental purpose of fisheries management is to ensure sustainable production over time and to promote the economic well-being of fishers and industries that rely on this production. Some of the biological parameters that go into stock assessments are growth, maturity, abundance indices, natural mortality, recruitment, and fecundity. Black rockfish are one of more than 60 species of rockfish in the Gulf of Alaska and California current ecosystems. In Alaska, they comprise 50 to 60% of the recreational landings. In Washington, 80% of the recreational landings. In Oregon, they bring in $20.6 million in annual revenue for the recreational fisheries alone. And in California, they comprise 15 to 30% of nearshore landings. There are currently 3 management areas and 4 assessment areas for black rockfish off the U.S. West Coast. The Alaska Department of Fish and Game is currently underway with the first black rockfish assessments in the southeast, north coast, and westward regions. We have a strong time series of age and maturity data from Oregon. However, the 2023 black rockfish assessment utilized data from different assessment areas, leading to Washington and California borrowing maturity estimates from Oregon. It is valuable to ensure that we are collecting data and managing fishing areas appropriately for the long-term benefit of the fishery and those that rely on them to lead to more sustainable and ecological outcomes. Using mismatched data for management purposes can lead to inaccuracy in fishery stock assessments and can cause negative impacts on communities that rely on this resource. Looking at a study from 2011 on California sheephead in Southern California, they were sampled to estimate age maturity off of the Channel Islands. Within this relatively small range, they did find differences in their age and length at maturity. And then they evaluated the effects on sustainable fishing yield if space was considered in the stock assessment model. Using spatially explicit information, they were able to increase sustainable yield by 21%. So seeing these differences in these traits in California sheephead off of Southern California and now incorporating black rockfish who exhibit a large natural range from the Aleutian Islands to Central California, we can infer that across this broad range where we sample them in even cooler waters in Alaska, we may also see differences in their age and maturity throughout this region. If a species encompasses a small geographic range and we observe differences in these key life history traits, we can infer we would see the same for species with such broad distributions. Spatial variation in life history traits can in part be explained by the temperature size rule, where ectotherms grow faster and reach smaller maximum sizes in warmer southern waters, while their counterparts in cooler northern latitudes grow slower and reach larger maximum sizes. So we are sampling in all 4 states across these major biogeographic regions indicated here. I am not sampling alone. I am working with many undergraduate students, other graduate students, volunteer anglers, and agencies to collect this data across this broad range. To estimate fish age, we use the break and burn method, where we break the otolith at the center and burn it over a flame and then observe the annulus rings under a microscope, similar to how one would estimate age for a tree. We then use the von Bertalanffy growth function to estimate size at age. For the purpose of this talk, we will only be focusing on female size at age. We see clear differences in region 1 and region 6 and overlap in 90% confidence intervals for indicating similarities between regions 2 and 3, 4, and 5. And because fish don't care about boundaries, but we do, we will also be looking at state-based results for females, and we can see a more clear latitudinal pattern of growth estimates here between states. We then estimated female maturity using histological techniques where we sent a cross-section of an ovary to be made into an histological slide and observe various maturity stages as seen here. We then used a logistic regression to estimate the proportion of mature fish and length and age at maturity. We also evaluated biological and functional maturity. Looking at functional maturity, which is the parameter currently used in stock assessments, and this indicates an individual that is capable of reproduction and will spawn in that given year, we see overlap in 90% confidence intervals, even though we do see differences between length and age at maturity, indicated by the dashed red line on these plots. So do we see spatial differences in these estimates? Another way to look at these results are plotting these estimates by latitude. We can see again that the difference between regions 1 and 6 for female size at age for both the theoretical maximum growth and growth coefficient, and we don't necessarily see a clear latitudinal pattern for maturity estimates, and this could be due to a smaller sample size, and we recommend an increase in sample size for future studies. So regional differences in life history traits can suggest separate stocks, but more information is needed, such as genetics and movement information. I would like to thank the Hatfield Marine Science Center Student Awards, specifically the Holtz-Krebin and Oregon Department of Fish and Wildlife Marine Reserves Award for supporting me through my master's thesis, and all of the volunteer anglers, agencies, biologists, and students that have helped with this project.
Thank you, Madison. We're going to do one more, and Wave, I might jump and do a live one in between if you're willing. Okay, so we're going to do one more recording with actually Madison's lab mate. We had Perry Geisen, who also worked with Cheryl Barnes, working on her master's, who also just recently graduated and completed her thesis. And she completed her work with the 25 Holt and Reynolds Awards.
Hi, my name is Perry Gerson, and today I'm going to be presenting results from my master's thesis, Food for Thought, using predators as samplers to understand prey availability throughout the Gulf of Alaska. Now, we're going to start by talking about marine heatwave impacts for the region. In this plot, we can see trends of Pacific cod abundance through time, where there's a red bar at the end of the time series representing during and after marine heatwave years. We can also overlay the number of humpback whale calves in the Gulf of Alaska on top of that Pacific cod abundance to really see how both of these predator populations experienced a sharp decline during and after the marine heatwave years. This decline was also seen in the common mirror, which experienced a mass mortality event during the marine heatwave. These predator declines are surmised to be in part due to a decrease in prey availability. That said, we are missing data on these important mid and lower trophic level species to really disentangle and understand climate impacts on marine predators. And so this is getting to the crux of my master's research, which is to get creative with pre-existing data sets and use diet data to understand prey trends for the Gulf of Alaska. When I say diet data, what I mean is that let's say we have fish 1 and fish 2, and they're caught in 2 different regions. We can then sample their stomach contents, and we can look at it and we can see that in the stomach of fish 1, we have 3 shrimps and a forage fish, and in the stomach of fish 2, there are 3 forage fish. We can then assign that stomach contents to our 2 different regions. So we can think of region 1 as having more shrimps and less forage fish, and region 2 as having more forage fish and no shrimps. We're not using diet data to make these overall abundance or biomass estimates for these prey groups, but we are using it to get at this idea of where there's relatively more and relatively less of these different prey groups. For my thesis, we focused in on trying to understand prey trends for 3 focal prey groups, including krill, shrimps, and forage fish, as they are commonly occurring in these predator diets and can account for a large proportion of their diet. Now, we're trying to understand these 3 prey groups using diet data from 3 groundfish predators, including arrowtooth flounder, Pacific cod, and walleye pollock. We narrowed in on these 3 predators because they are regularly sampled during the NOAA bottom trawl survey. Now that we have our diet data, we're going to generate these estimates of prey using models. And what we did is we built a model for each unique predator-prey combination. So we built a model for krill using diet data from arrowtooth flounder. We built a model for krill using diet data from Pacific cod, and we built a third model for krill using diet data from walleye pollock. And we did this for all 3 prey groups. So we did it for shrimps and forage fishes as well. And here's a map generated from these models. And what we can see here is we have categorized the different levels of prey. So in our yellow areas, we're calling those prey hotspots. So these areas have the highest levels of prey of this prey group for the region. And as we go down, we can see that dark blue has some prey, but not as much as our teal, green, and yellow spots. And we can create these maps for shrimps as well, where we can see that nice big hot spot in the central GOA and some smaller hot spots throughout. And we, of course, generated maps for our forage fishes group as well. And again, we see that nice big hot spot in the central GOA in the mouth of Cook Inlet, and another sizable hot spot towards the eastern GOA. Now, I'm going to leave us with a few take-home messages from my work. First and foremost, I hope my work exemplifies that we can use diet data to map and better understand trends of our data-limited lower and mid-trophic level species. And I would like to pose this question of what other creative uses can we come up with for pre-existing data sets? Thank you, and I would like to extend my gratitude towards my funders, include the Lillian Brucefield Reynolds Scholarship and the Curtis and Isabella Holt Educational Award. Thank you.
Thank you very much. We're going to jump into, split up our recordings here just a little bit because Wave is willing to be sacrificed forward. So Wave Moretto is working on her PhD in Fisheries, Wildlife, and Conservation with Scott Appel, and her work was funded with the 24 Markham Award.
Can everyone hear me okay? Hi everyone, my name is Wave. I'm super excited to be here today talking about my ongoing research from divers to DNA, connecting fish, kelp, and coastal change. I'm a PhD student in Dr. Scott Appel's lab, and this work was funded by the Markham Award and continues to be funded by other Hatfield Awards. So I study Oregon's kelp forests, which are dominated by bull kelp, which is a canopy-forming species that lives in cold water reefs. And our kelp forests are dominated by rockfish, which are in the genus Sebastes, and you can see swimming around here. It's also important to note that kelp forests here are annual, so bull kelp peaks in abundance during the summer or early fall. It naturally dies off during the winter, and then it starts to grow again during the late spring to early summer, and that creates this cyclical growing cycle. I also wanted to highlight this really important trophic relationship in Oregon's kelp forests. Bull kelp is predominantly eaten by purple sea urchins, and these are kept in check by predatory sea stars or sunflower sea stars, Pycnopodia, which prevent overgrazing. But in 2013, we had the onset of sea star wasting syndrome, which resulted in massive declines of sunflower sea stars and huge increases in purple urchin populations along our coastline. And so this allowed them to graze freely on kelp forests. And so this predation pressure was then also coupled with heat stress from the 2014 warm blob and 2019 marine heat wave. And so this combination of increased temperature and predation did not bode well for our kelp forests, and we saw this shift to urchin barren systems. And this loss of kelp on our coast was quantified by the Oregon Kelp Alliance. They present this really alarming statistic that from 2010 to 2022, Oregon's peak annual kelp canopy has declined by about 70%. And so my research is focused on how kelp forest change is influencing fish populations, as Oregon's kelp forests support many fishery species like black rockfish, lingcod, and salmon, but we don't have a lot of data specifically on kelp forest fish for this region. And so my specific goals are to optimize methods to study Oregon's kelp forest fish communities and to document the status of these communities to establish a baseline in the face of future change. And so to do that, we are scuba diving at 3 different locations on the Oregon coast, Cape Foulweather, Cape Arago, and Port Orford on a quarterly basis, and we started this in the summer of 2024. We dive within a kelp forest and an urchin barren, and we do a bunch of different survey techniques. So we do these belt transect surveys where one diver is operating stereo video and the other is doing an underwater visual fish count. We get data on fish abundance, canopy and understory kelps, key invertebrates, and other habitat characteristics. And stereo video works. The diver films a fish with left and right side cameras, and then we upload those videos into a program called Event Measure, which gives us total length measurements for the fish without ever having to catch or handle them in any way. We just have to film them. And so far, we've detected significantly more fish using that stereo video method relative to an underwater visual count. And we found that kelp canopy, habitat structure, and temperature all significantly influence kelp forest fish diversity. We have also seen significantly less rockfish on reefs during the winter kelp forest decline, and significantly more and significantly larger rockfish in our northernmost kelp forest site relative to urchin barrens. And so these findings have been really exciting, but there are still a lot of limitations, specifically in our ability to detect fish diversity, as we're seeing relatively low species richness across both scuba methods. We see a lot of our dominant species like black rockfish, but less of our cryptic or more seasonal species. And so a solution for this is to use environmental DNA, or eDNA, which is what fish or any organism sheds into the water as it swims. We collect a water sample, and this provides us with a way to look at species diversity without ever having to actually physically see that organism. But this is still limited to our ability to be able to get out in the water and get a boat on a good day to dive. And so we're looking for a way to be able to continuously monitor those ecosystems. And so that brings me to the next phase of my research, working to further develop an autonomous eDNA sampler that was first developed by Genevieve here in the iLab at Hatfield. So this will filter seawater and preserve it in situ, and then it can be serviced by a diver, and so we can get these samples more regularly. And I wanted to highlight that commercial counterparts can cost up to $50,000, but this sampler will cost about $500 to $1,000, and it will provide the first continuous data set for fish communities on the Oregon coast. And so with that, I'd like to thank everyone who's made this project possible so far, especially scientific divers Alexander Bayless and Olivia Boyson, and the rest of the Happel Lab. And I really want to thank the funding from the Markham Award, as that allowed me to jumpstart this project. So thank you.
Thank you very much, Wave. Appreciate it. Gonna do a little photo shoot here. And so we're gonna jump back into a recording, Marnie, with Riley Boyd. They completed their master's with Fisheries, Wildlife, and Conservation with Taylor Chapel, with the support of a 25 Jones Award. Cameron, would you be willing to? Okay. Poor Riley has broke the computer. We'll try to get that one back going again here in a second. But Cameron Royer works with Sue Spinagle, who you've heard a little bit about already today. He is completing his master's in integrative biology, and he also has some support currently from a 2024 Markham Award. And we'll see if we can pivot here in our computer programming.
Hi everyone, I'm Cameron. Today I'll share part of my dissertation supported by the 2024 Mamie Markham Award, using a combination of genetics and high-resolution imaging to investigate species-specific larval rockfish distributions in the Northern California current. Rockfish are, do I need to push go? It's not, no. Timeout.
Everybody, some patience here with our computer.
It's definitely been more than 15 seconds.
Seconds are clicking down. You can try.
Okay, I'll just go through. So rockfish are foundational to Oregon's coastal communities. They support valuable fisheries, play important ecological roles, and hold cultural significance for indigenous peoples along the Oregon coast. So understanding how larval fish survive and eventually replenish adult populations is critical to communities and ecosystems that rely on healthy rockfish populations. Like many marine fishes, rockfishes occupy very different habitats throughout their lives. Adults often live near reefs and other rocky habitats, while larvae spend weeks to months developing offshore in the open ocean. And conditions experienced during the larval stage can strongly influence survival and future population size. So understanding these processes, to understand these processes, we need to know where different rockfish species occur and how their distributions relate to oceanographic features. Many rockfish larvae are notoriously difficult to distinguish visually, even with established morphological keys. There are limits to how far we can go without genetics, which makes it difficult to resolve these species-specific distributions I'm after. The Northern California current is also a highly dynamic system. Currents, fronts, and plankton communities shift quickly, while at the same time traditional nets integrate organisms across the volume filtered, smoothing over fine-scale spatial structure and larval distributions. So to address these challenges, we used 4 years of winter and summer surveys spanning Northern California to Washington. And these cruises combined depth-discrete net sampling and high-resolution in situ imaging across broad spatial and temporal scales. One key tool we used is the MOCNES, a multi-net system that opens and closes at 25-meter intervals. And this lets us collect larvae from discrete depth layers, providing vertical resolution that traditional single-net tows cannot. Larvae are patchily distributed throughout the water column. They can be aggregated around vertical gradients in temperature, prey, and currents, and may occupy distinct depth layers. And so having that depth discrete biological sample to identify where species occur is crucial to understanding vertical larval distributions. For this study, I focused on larvae collected in the upper 50 meters of the water column. These fish were sorted from nets, measured, and prepared for genetic analysis to build a depth-resolved data set of larval size and species identity. So across all cruises, we measured more than 13,000 larval rockfish, and this size distribution here captures a broad range of larval development stages across seasons and years, but does not resolve species-level differences in early life history. So in collaboration with the State Fisheries Genomics Lab at Hatfield Marine Science Center here, we genetically identified larvae using a panel of nuclear microhaplotype markers. So far, we've identified about 1,000 individuals, but there are thousands more currently being processed. With those species identities assigned, we can examine how species distributions changed across space and time. But even with just a subset of the samples completed, these results show that species composition can vary substantially among stations within a single cruise. These data also reveal clear differences in size distributions among species, and once all the samples are identified across cruises, we'll be able to examine how these distributions change across time and space. However, species identities and MOCNES samples alone do not provide the fine-scale spatial resolution needed to fully understand what drives these larval distributions. To do that, we used ISIS, the in situ ichthyoplankton imaging system, which captures high-resolution images of plankton in their natural habitat. So as ISIS moves through the water column in a TOYO pattern, it takes a continuous image across the entire transect, which preserves natural spatial structure of larval fish distributions across the volume filtered, while also collecting environmental data. So ISIS provides an incredibly detailed view of larval distributions and the environmental conditions they experience. But the challenge with ISIS is that it can't distinguish between larval rockfish species, and so it can only generate these distributions at the genus level rather than for individual species. But that's where our MOCNES collections become important. MOCNES and genetics tell us which species are present and where they are generally, and while ISIS shows where those larvae occur and the conditions they experienced. So by combining MOCNES, genetics, and ISIS, we can link species identities to these fine-scale spatial distributions, and that will allow us to move beyond asking where larval rockfish occur and begin asking why different species occupy different parts of the ocean. This work would not have been possible without the support of our collaborators, ship crews, Plankton Ecology Lab, State Fisheries Genomics Lab, and funding support from the 2024 Mamie Markham Award, as well as the National Science Foundation. Thanks.
Thank you, Cameron. All right, while we take our little photo break here, I just want to remind everybody that we should be voting on our favorite talk. So if you do not have a ballot and you need one, raise your hand and I'll get it to you. Okay, we'll get a couple back there. Do you want to kind of pass those around? Okay, so some ballots are coming out. I want to make sure everyone gets a chance to get an award if they deserve that, which you all do, which is great. We're going to try another recording. Angelina Zullo graduated recently as well with her, or completed her project, PhD in integrated biology with Sarah Hinkle, also supported with a 2024 Markham Award. Everybody cross their fingers. We'll see if this works. How we doing, Marnie?
Okay. Hi, everyone. My name is Angelina Zillo, and I'm a PhD graduate from the Henkel Lab at Hatfield. I'm so sorry I couldn't be there today. I'm at the Phycological Society of America conference in Alaska. But today I'm going to be talking to you about kelps and surf grasses. So, just to bring up the classic foundational term, foundation species, which means organisms that provide habitat resources that support diversity. Of course, we're with the classic example, redwoods, and then we have our underwater forests, and that's what I'm going to be focused on today. Here's some classic headlines that we've been seeing in recent years. For instance, kelp forests are vanishing along California's coast. We're seeing declines in the Puget Sound. And then there's just this overall arching of the vanishing forest. But there is some evidence that there are some glimmers of hope in other places, like here in Oregon and in Mexico. Of course, we're seeing unprecedented declines in other places like Monterey, but maybe not the case in other parts, like here in Oregon. And so today I'm going to be talking to you about Egregia menziesii, commonly referred to as the feather boa kelp. This is considered a foundation species, and it's a perennial, so it can live for many years. I'll also be talking to you about Phallospadix, which is an angiosperm, pictured here on the right. This is a surf grass, so this is not a kelp, but it is very common in the intertidal here in Oregon, and it is also a foundational species and also can live for many years. And so I'm going to be going over quite a bit of topics today, but I just want to show you some pictures of why I picked these two species to study, but essentially they overlap quite a bit in the intertidal in Oregon, and they both are dominant canopy formers. So I kind of want to examine this relationship and just their roles here in Oregon. And so I looked at like many different things, included rapid warming, canopy disturbance, diversity, and understory. And I'm just going to be briefly going through all of that today. And so in the lab, here's an example of what the lab setup looks like. We did these experiments in the experimental seawater facility here at Hatfield. And you can see that basically we were able to set up all these different tanks. We put them at different temperatures. We did different light treatments, different nutrient treatments, and basically did a bunch of different things to them. And this is what they look like when they're trapped in their bottles. But basically that's an Egregia individual on the left. We have a Phallospadix in the middle. And then we also did a field experiment where we looked at them side by side. So you can just see all of that. And so here's an example of a happy, healthy Egregia from straight out of the field. And that's just to show you what they look like when they're happy. And this next slide will show you what they look like when they're not happy at some higher temperatures. And so you can see by these images, especially by that bottom image, if they're put at too high of temperatures, they will essentially disintegrate in their tanks. And then on the right, you can see that the tissue is starting to decay. I don't have any pictures like this for Phallospadix because Phallospadix did not really have a strong response to temperature. So overall, the kelps don't do well with rapid warming, but surfgrass does just fine. So now I'll talk about canopy disturbance. And so for this, we did quite a bit. We did a few years of field experiments, and we did this at 4 sites here in Oregon. We had 2 central sites, and then we had like a mid-central south site, and then we had a southern site. And we did these long transects and we tracked the canopy over time. And what I'm going to show you here on the left, this is a picture of a healthy, nice Egregia bed at Boiler Bay when we first started this experiment. And on the right is that same exact bed 2 years later. You can see Egregia was missing. And this was pretty strong trend. We saw quite a bit of kelp disappear at our sites, and surf grass remained pretty strong. And then for diversity and richness, I'm just briefly going to show you an example of what we talk about or what we mean when we mean diversity in the intertidal. It can include quite a bit of different organisms, including algae species, but we did not see differences in diversity and richness between egregia and surfgrass. So overall, the diversity and the richness was the same. But when you look at the individuals that are underneath these canopies, you can see that there's a pretty big difference. Egregia houses a lot more reds, Phallospadix houses more invertebrates and more coralline algae. So overall, diversity and richness was the same between the two groups, but when you look at the individuals, the reds and the coralline algae kind of separate between these two groups. And so the main takeaway I want you to take away from this is that, of course, we're seeing quite a bit of losses of kelp, especially in these big headlines, but a loss of kelp does not equal a loss of biodiversity. We will, of course, see community shifts as we shift in dominant canopy, but as long as there is a dominant canopy, the community will persist. And then, of course, I'll acknowledge my Markham Awards that I got for 2022 and 2024 that helped all this research move forward. I'll thank all my other sources, and then I'll also thank my advisor, Sarah Henkel. Thank you so much for listening.
All right. We're going to continue with our next on-site presentation with Alexander Bailis, who's working on a PhD in CEOS with Maria Kavanaugh, also supported by a 24 Markham Award.
Thank you. Yeah, we'll see. I have faith. Yeah, I think so. Sweet, thank you. Hi, my name is Alexander Bailis. I'm a graduate student here and excited to share with you today about what we've been doing the past few years for our Markham Award. We've had a great time, we've learned a lot and have a lot of fun, and so thank you all for being here, and without further ado. So, our project really wanted to understand the structure and function of Oregon's nearshore marine ecosystems. And the lens with which we wanted to study these ecosystems was using phytoplankton. If you can understand the size distribution and the community composition of the phytoplankton, you have a good first order assessment of what's going on with the zooplankton and the fishes. And so the way that which we were going to do this was starting with these sensors that were homemade, we made in-house to look at the phytoplankton. We were going to deploy these in kelp forests, seagrass beds, estuaries, and use these to kind of get a general assessment of what's going on in this ecosystem. And so starting at the beginning with the sensors, the initial design we had was out of PVC tubes. We made these in undergrad. It was a beam that would fire through and look at the turbidity, giving you an idea about the suspended materials therein. However, with some great collaboration with the iLab, we made something much nicer on the other side. 3D printed design. It's got 3 light sensors inside. Total part cost is $150, making it 2 orders of magnitude more cost effective than commercial analogs. Furthermore, we changed it to an above water design. You can put this on a boat, on a mooring, and so you don't need to be a scuba diver to service it, and it has basically no biofouling. We've also made it in a way where there's no soldering, meaning if you can put together Legos, you can build this sensor. A couple other labs have adopted it, and it's also done a full transatlantic, trans-Pacific, and 3 tours along the Oregon coast. So very excited about it. But the sensor is measuring the phytoplankton, right? And so going back to that, these are 4 of the most dominant phytoplankton that we have here in Oregon. And we wanted to measure kind of the signal that we get from those to see what our sensor is going to be looking at. Ground truth that, if you will. But one of these is not like the other, and luckily we had somebody prime us on harmful algal blooms already. This is Pseudonychia. Pseudonychia is our most dominant harmful algal bloom here in Oregon, produces a very potent neurotoxin. The whale skeleton that we actually just got was found washed up on a beach with domoic acid in its tissues. Very dangerous for marine ecosystems and also for economies, because it shut down our fisheries. So we devised this experiment where we took those 4 most abundant phytoplankton in our system and we grew them in cultures. And we grew them in cultures so that we could look at their kind of spectral signature, or how they change the color of the water, to know what our sensor is going to be looking at. And we measured them with a bunch of very fancy high-tech instruments and found something very exciting that we absolutely did not expect. What we found was that Pseudonychia, that harmful algal bloom species, actually had a very unique signature. Right here on the top, this is the ocean color fingerprint of Pseudonychia, basically how it changed the color of the water. And you see it kind of has a distinct M shape, whereas at the bottom it's the Lasiosira, a benign phytoplankton, and it has kind of a flat top. While our eyes can't tell the difference between the spectral signature, the sensors that we'll be making can, and actually the new NASA PACE satellite that was just launched last year can also discern between these two spectral signatures, which is really, really exciting for us and something we absolutely did not expect. This work has been submitted to a Journal of Harmful Algae with the goal that when the satellite passes over in a day, we'll get an image of the whole ocean, and from those color pixels, we can kind of pull out and see whether a toxic harmful algal bloom is present or more benign phytoplankton that are good for the ecosystem and for our marine economies. So this is very exciting, but also meant we had to go back to the drawing board and make our sensor better. Our initial sensor that was $150 could not detect the pseudo-Nietzsche signal. And so with some great work at the iLab, cannot thank these guys enough. I mean, super, super helpful. We made a new sensor that can actually look for the pseudo-Nietzsche signal, detect these harmful algal blooms. And in a cruise that we did recently, thank you Wave so much for help with this, we were out in the Monterey Bay, detected a big pseudo-Nietzsche bloom, those are those red dots, and found that indeed this sensor could actually pick up that signal. So what we found in the lab was proved in the field, and that was very, very exciting. Thank you so much, Jacob Drummond, everyone in the iLab. And so to quickly summarize, we found that our most dominant harmful algal bloom species here in Oregon has a unique spectral signature, changes the water in a special way that we can identify with this new sensor that we have made and with the new NASA satellite. We've made two different sensor designs. One is very low cost, can look at phytoplankton, pollution, water quality. We're working to get these mounted on fishermen's boats as well as at aquaculture farms. And this new prototype that we're still working with the iLab on that can look for this pseudo-Nietzsche signal. And all of this goes into a larger project, which you've heard from both Cameron Royer and Wave's presentations, where we're looking at how this energy and carbon from these phytoplankton are moving up the ecosystem. Concurrently with their dive surveys, we're sampling for phytoplankton and seeing how the size and distribution, community composition of the phytoplankton is affecting things like the fishes in our nearshore fisheries. And we just want to say thank you so much. You know, absolutely none of this work would be possible without you guys and without our generous donors, especially not without the iLab. They've been absolutely instrumental in this work and so accommodating and able to really help us in any and every way. We thank our co-authors at the CCAP Ecology Lab, Phytoplankton Ecophysiology Lab, and of course, the wonderful people here at Hatfield that we've had the pleasure of collaborating with. So thank you so much. I only have 5 minutes. If you ever want to chat about this, I can talk for hours. So please come find me. Thank you.
All right, that was amazing. Thank you very much, Alexander. Our next presenter is Jessica Schultz. She's also a recording as I fall over here. She was hoping to be back in time, but they're stuck in the field in the Puget Sound. She's working with Taylor Chappell, working on her PhD, and she got some funding from a 25 McNeil Award. So we'll see if we can get the recording started.
Hi everyone, my name is Jess Schulte and I am a fifth-year PhD candidate in the Chapel Big Fish Lab here at OSU. And I am very sorry I can't be there with you today. I'm currently out in the field, but I am excited to talk to you about some of my ongoing research looking at broad-nosed seven-gill movement and genetic connectivity here in the Northeast Pacific. So seven-gills are large apex predators found in temperate coastal ecosystems around the world. They're generalist and opportunistic feeders and are considered apex predators and important species in the ecosystems they're found in. Here in the Northeast Pacific, seven-gills range from southern Alaska down to the Baja Peninsula, and they do these long annual migrations up and down the coast back to specific bays and areas where they're kind of seen in sort of hot spots. And so those hot spots include 4 main locations. All the way down south, we've got San Diego, and then up north, we've got San Francisco, where seven-gills are caught as targeted recreational fishing. And then we've got Willapa Bay, where a previous study had been done on them, and then just recently up in Puget Sound. So over to my study sites, Willapa Bay and Puget Sound, which are 2 large estuaries home to important commercial fisheries and managed species, and also home to established receiver arrays. So that means that tagged animals with acoustic tags can be heard on these established arrays. So basically it helps us track animals as they swim throughout the bay so we can fine scale track their movement. And it also allows us to track them as they leave the bay. So Willapa Bay is part of NPACT, which is a broader Northeast Pacific acoustic array that allows us to track animals as they leave the bay and swim up or down the west coast and come back. So to understand more about these sharks, I've been going out 2 to 7 days a month and I've been collecting measurements. I've been deploying acoustic tags to help track that movement, and I've also been collecting tissue samples such as genetics. So over the last 4 years, I have sampled over 340 individual sharks with just 12 recaptures, and then I have deployed over 80 different acoustic tags on individuals, capturing different size, class, and sexes. Looking into the acoustic detections, the upper third indicates 7 gills tagged in Puget Sound, with the rest being tagged in Willapa. You'll notice that there doesn't appear to be a lot of individual movement between these systems, so they appear to stay separated during those summer months, which led me to the question, are they all one population or are they completely separate? They don't seem to be swimming back and forth between these bays, and so what does that mean for the broader population? So to further complicate things, there was a previous study done in 2015 which used a related species, 6 gills, as a reference species to study 7 gills. And they found that 7 gills in Willapa Bay in San Francisco were 2 separate populations, which led me to the bigger question of, we have all of these locations that are close to each other. How are these individuals part of separate populations? So I decided to look into that using SNP genetics data. So to get samples from each of those locations, I reached out to collaborators down in San Diego, as well as in San Francisco, Megan Holst, as well as researchers down in Southern California, who happily provided samples that I was able to analyze for genetics. So I used the Center for Quantitative Life Sciences here at OSU, who helped me analyze these samples and use bioinformatics and filtering to process them. So here's the results, very exciting. This is a PCA plot. Each dot represents one shark, and the colors represent the four different sampling locations. You'll notice that there's no pattern here at all. You would see clustering if they were separate populations. So basically, what we're seeing is that these are all one population. This is the FST analysis that I did, and this tells the same story with actual numbers, showing that it's all one broader population. But that reminded me of that 6 gill study that found separate subpopulations. So I went through and did another analysis where I did extra filtered results to make sure that there wasn't any underlying artifacts in there, and I got the same exact results as a robustness check. So what does this mean? All one population, very exciting. Up next, I need to calculate the effective population size. If we've got one population on the West Coast, we need to understand how big that population is, especially given how important the species might be on the Pacific Coast. I just want to thank Hatfield and the McNeil Fund for funding this research. I wouldn't be able to do it without them as supporters, as well as all of the collaborators and partners that have been involved in this project. Thank you so much.
And our next speaker is also up in Puget Sound with Jessica. Ethan is there, and he is working on his PhD in fisheries, wildlife, and conservation with Taylor Chapel, and was funded in 2005 with an ODF&W award.
Hi, my name is Ethan Personius, and today I'm going to be talking about the occurrence, relatedness, and movement of soupfin sharks in Willapa Bay, Washington. This is just a small part of my overall PhD research on the movement, foraging ecology, and population genetics of soupfin sharks in the Northeast Pacific. The soupfin shark is a critically endangered species with several genetically and geographically distinct populations around the globe. In the Northeast Pacific, their range extends from British Columbia to south of Baja California. Throughout the first half of the 20th century, this population experienced significant fishing pressure buoyed by continually increasing demand for liver oil. Despite adopting more effective gear types, this fishery saw declining catch per unit effort until its eventual collapse. One of the most interesting patterns from the historical catch data is that the sex ratio changes dramatically by latitude, with female-dominated landings occurring off Southern California and males dominating the catch in higher latitudes, like in the waters off Washington State where the majority of my sampling effort is focused. And that seems to align with our catch data from Washington, where out of 51 sharks sampled from my research, only 3 have been female. Over the last 2 sampling seasons, we captured 7 individuals between 42 and 67 centimeters total length in a primarily adult male-dominated aggregation site. Here you can see a visible umbilical scar on a 44 centimeter individual. Given that young of year sharks have never been recorded north of Tomales Bay, California, we wanted to first determine whether these individuals represented multiple litters during the same pupping season, and 2, we wanted to compare the estuary departure time between age classes and determine the drivers of habitat selection for young of year soupfin sharks. To do this, from each of the 7 young of year soupfin sharks captured, we collected a fin clip and muscle biopsy. Then a subset of 4 individuals were fitted with internal acoustic transmitting tags, and their movements within the estuary were monitored until their departure in the late fall. For these sharks, we estimated age at capture using the Macmillan 2021 growth curve and the most commonly reported maximum embryo size from Ripley 1946. 5 of the 7 sharks were well under 1 year of age, and the 2 sharks exceeding 60 centimeters in length are right around the 1 year mark, possibly returning to the estuary. 6 of the 7 young of year and near yearlings were included on a single nucleotide polymorphism panel focused on broader Northeast Pacific genetic connectivity. I filtered a set of genetic markers and conducted a colony SIBSHIP analysis and concluded that of our sample, we contained no full or half siblings. Genetic and size-structured catch data indicate that young of year soupfin sharks in Willapa represented 7 distinct litters across 3 pupping seasons. In other populations, this size class can grow rapidly before dispersing in late fall. Together, these patterns suggest that annual pupping occurs near or around Willapa Bay. Within Willapa Bay, WDFW maintains an array of 10 acoustic receiver stations. These receiver stations have a detection range of approximately 500 meters. Coverage is not perfect, but it does effectively represent the estuary gradient. To investigate within estuary residency, we compared departures of young of year and adult soupfin sharks. Median departure date of the young of year sharks occurred 52 days later than that of adult sharks, suggesting that they continue to utilize the estuary long after the adults have already left. I utilized the high-resolution Willapa Bay submodel of the broader Live Ocean model to get daily spatially explicit averages of salinity, temperature, oxygen, and nitrogen at each acoustic receiver station on each day of the monitoring period. To model shark presence, I created a presence-absence matrix for the length of the monitoring period, where each shark was deemed present if it was detected at a station on that day, and absent if detected in the bay, but not at that particular acoustic receiver station. Before model selection, I checked correlations among covariates to avoid including strongly correlated environmental variables in the same model. Candidate GAMs were compared using AIC. Final structure retained salinity, phytoplankton, and receiver station as a random effect. Young of year presence had a strong negative relationship with salinity and a nonlinear relationship with biological nitrogen, suggesting greater use of estuarine rather than marine conditions within Willapa Bay. However, receiver station was the strongest predictor, likely reflecting habitat features not included in the model. Next steps for this work will focus on including the 2025 detection data in this model once environmental data from that period become available. I will also model the drivers of adult soupfin shark use of these estuary systems and improve habitat classifications within the detection range of each receiver to include in future models. Finally, I would like to give a huge thanks to everyone who's helped out with this work and to the ODFW Marine Reserves and Markham Awards for supporting this research. Thanks.
All right, so our next speaker is Joa Villapiera, who is working on a PhD in fisheries, wildlife, and conservation with Mauricio Cantor and received a 25 Pratt Award.
Thank you. Check. Can you hear me? Well. Okay, so thanks guys. I'm going to show you a little bit of, or as a part of my work on how we catch dolphins feeding, but not from a boat, actually from space. Ooh. It's not that crazy, but it's actually a culture we can see from space. So these bottlenose dolphins, they do something almost no other population of dolphin does to catch fish, actually. And it's learned, and it's local, and it leaves a mark. Sounds very poetic when I say it in the mic, but as you can see, they swim in a tight circle, throwing up a curtain of mud, and then the fish jumps, and the group feeds, and it's actually very coordinated, and it's specialized, right? So this is basically the whole talk in one line. The behavior writes on a signature into the water, and if we can read that signature, we can find the behavior. So, but the big problem is we can't be in the right 50 meters, in the right 5 minutes across almost 2,000 kilometers square in one place at the same time. It's like trying to find a needle in a very, very wet haystack. But a boat survey, you can think of it like a soda straw, a metal soda straw, like a narrow field of view, and you have an event if you're lucky, and you have to be there as well. So, I mean, I would love to be on a boat right now, but so flip it. So we go from overhead, and I mean way overhead. The whole bay is at frame at once, and every trace is just sitting there, kind of waiting to be tracked. And Eric Ramos and colleagues showed that the traces are there and that it is possible to find them and count them. But it's still a huge area. It's still the whole Florida Bay. So my job here was trying to make this finding kind of more automatic and more scalable. So the idea here was to train a machine learning model, like those models that identify faces or cars, and then retrain this model to find the mud ring traces. Okay, and this is actually a feature. It's not just a shortcut, because first we started training this model on free available government imagery, so anyone could reproduce our work anywhere and with their own study systems, and yeah. So the rings, coming back to the rings, they are not random smudges. They have consistent sizes, and they have consistent spacings between them, which is exactly what a detector like this needs to lock onto. And they actually swirl. So most tracers are these nail-like spirals, and cataloging these shapes told us what to label and what to ignore. And that's because these kinds of models, they work on examples, right? I label the rings, and as importantly as labeling the rings, we label the lookalikes of the rings, so the model kind of knows what not to flag as well. And YOLO, the software framework we use for that, we kind of just throw an image there, jet there, and then it returns every detection in a single pass, so it's fast enough to run across the whole entire scenes of Florida Bay. So you hand it a fresh imagery, it lights up the real rings it never has seen before, and this is kind of the moment the idea becomes a tool, right? And you can start asking more deeper ecological questions or other biological questions. So we started by testing with freely available data, and now with the help of the Evan Pratt Memorial Scholarship, we're using sharper imagery to answer deeper questions as well. And these questions could be like where, how often, whether this specialized static is changing over the years, and it's basically going from these scatter anecdotes to a map like that, for example. And now taking a step back to the big idea, this is animal culture, right? So this imagery archive, it's kind of going back decades and becomes a record of how this learned behavior is changing over time, and it moves through a population. And that's basically the roadmap of the project, how the project was going. So free data answers kind of the where, better data answers the when and why, and the densest data set kind of answers how the behavior spread or how it's shaped by sea level rise. And yeah, thanks for the opportunity here. Thanks, Mauricio and Lee. And this work is in Even Pratt, of course, and this work is in honor in memory of I'm going to show him again, of my friend Eric Ramos. That's it. Thank you. Oh, it was like that? Why?
All right. Luke, are you ready? Luc Bobet is our next speaker, working on his PhD in integrative biology with Sue Spinagle. Work was funded with a 25 WIC award.
All right. Yep. All right. Yeah, so what I'm presenting today are some results from the first chapter of my dissertation. These results are still in progress, so subject to change. But yes, they were funded in part by a 2025 WIC award, and they focus on anchovy, which serve as an important link between plankton and upper trophic levels in marine ecosystems. Anchovy are short-lived, so their abundance varies a lot between years. And off the west coast of North America, they exist in 3 distinct spawning stocks. The northern stock is the one that I'm studying, and the central stock is the one that has historically been more studied, and it's where the research that inspired this work was done. And this is a time series of adult anchovy abundance in the central stock. It shows that it is highly variable across years, and the time frame highlighted in yellow is a bust period when anchovy abundance declined. And being able to predict bust periods like this could be very useful for managing anchovy and the species that rely on them. And this coincided with an increase in the trophic position of anchovy larvae, which could be a useful indicator of those declines in abundance. But in order to use this as an indicator, we need to understand the mechanism that drives that relationship. So the authors of this previous study proposed that it was the size of the phytoplankton cells that influenced how much energy was routed through an intermediate heterotrophic protist trophic level, and ultimately reached the anchovy larvae at varying trophic positions, which influenced their survival to adulthood. I'd expect that during those conditions where phytoplankton cells are larger, there will also be higher primary productivity, resulting in more prey available for the anchovy larvae to feed on, also reducing their survival. So these are 2 alternative mechanisms that could drive this relationship. And in order to try to tease apart those relationships, my lab sampled the planktonic food web on a very fine scale, and I was able to measure the performance of individual anchovy larvae from their muscular height at anus, a measure of condition, how much food is available relative to their metabolic demand, and also from their recent growth from their otoliths. And I measured their trophic position using the nitrogen isotope ratios in amino acids in their tissues. The relative abundance of nitrogen 15 to nitrogen 14 tends to increase as you move across successive trophic levels. And when we look at these in individual amino acids, some amino acids do fractionate in their nitrogen isotope composition, while others don't. And by comparing those 2 classes of amino acids, you can estimate the trophic position of individual anchovy larvae. And now for some results, I'm going to build a path analysis or path diagram for a Bayesian structural equation model that I used to examine the relationships among the measured variables. And here the arrows indicate those effects. The red arrows are negative relationships, blue arrows are positive effects. The values next to the arrows are the linear regression coefficient, and inside of the parentheses is the probability that that coefficient is different from 0. So first you see those arrows for age, which has an effect on these measured variables. Temperature had a negative effect on copepod prey abundance, also a negative effect on the phytoplankton cell diameter and on the normalized biomass size spectrum slope, which is an indicator of the efficiency of energy transfer through the planktonic food chain. Nitrate concentration had a positive effect on the phytoplankton cell diameter, and the effect of copepod abundance and the NBSS slope on those measures of anchovy larvae performance were not significant, but the effect of phytoplankton size on trophic position was. And finally, we had negative effects of trophic position on recent growth and muscular height at anus. So taken together, these results seem to indicate that it is trophic position that's relevant to the performance of these anchovy larvae, but not by the trophic transfer efficiency mechanism that I expected, or the copepod prey abundance that could serve as an alternative explanation. So more work is coming for that. And I'd like to thank the OSU Stable Isotope Collaboratory, which is where I did the GCC IRMS analysis to estimate trophic position. So if you would like to do that analysis with carbon or nitrogen isotopes and amino acids, you can contact them or me to find out what's involved. So thanks.
All right, Elena, I'm going to torture you. We're going to try Riley's one more time just to see if we can get that to work, if that's all right. Okay. All right, everyone, we're going to try that video just one more time to see if that works, so be patient with us. All right, Riley, if you're online, we'll figure out how to clip your video into the final recording for today's event. We're sorry so much. Okay, you're back up. Elena Corsa is also working with Sue Spinagle on a PhD in integrative biology and was also funded by a WIC award.
Not 13. Lucky number.
Lucky 13. All right, all set?
Why not? All right, everybody, thank you for hanging around here until the very end. I'm excited to share some of my PhD dissertation work looking at the growth and feeding of larval English sole. And for this, I received support from a Bill Wick Marine Fisheries Award. The larval stage of marine fishes is a vulnerable planktonic stage which represents a critical period for population replenishment. Relatively small changes in survivorship during this stage can have dramatic consequences for the population. Larval growth has been linked to survivorship and can be related directly to environmental conditions such as temperature, which can increase metabolic rate. It can also be related to how much and what the larvae are eating. The environment, however, can also set the stage per se, altering abundances of predators and prey in the vicinity of the larvae, which can then affect how much they're eating and affect their growth. I'm going to be exploring some of these relationships in English sole, which is an abundant flatfish that spawns during the winter. The data I'm using are coming from a series of 4 cruises over 4 years on 4 transects. Fixed stations were sampled with a depth discrete net, while an in situ plankton imager was sampled continuously cross-shelf. From the larvae in the nets, I used otoliths to calculate their recent growth and inspected their gut contents to see what they were eating. From the imagery, I was able to estimate the abundance of prey and predators in the same locations. So if we take a look at the distribution of the larvae, that was lagged, they're most abundant at the most near-shore stations, which I'm going to call Station 1, although there was considerable variability interannually and also spatially. So now let's take a look at what they're eating. And in their gut contents across all stages,
of development, years, transects, and locations, they are chowing down on appendicularians, which are small pelagic tunicates that are really cool, and if you're not familiar, you should learn more about them. So let's look at the relationship between the size of the appendicularians consumed and the fish. So this relationship scales very quickly in pre-competency stages where fish are gape limited and can't swim well, while when they're older, they're able to consume a wide range of prey. Interestingly, we detected station level differences, and fish at inshore stations were consuming appendicularians about 0.5 millimeter longer, which may not seem big, but it is huge when you are a 10 millimeter fish. So continuing with this idea of station level variability, we also found recent growth was generally higher at nearshore stations, although the magnitude of this relationship varied spatially and interannually, and there was a notable exception at our southernmost transect, Hesita Head. So now that we've covered kind of some background and foundational knowledge and looked at general patterns, let's dig into this direct and indirect ideas again. And for this, I'm going to use a structural equations model similar to what Luke presented, so thanks for that intro, Luke. So this is clearly a lot more complicated than that simple conceptual diagram, but we have our environmental conditions predicting predator and prey abundances, which are then allowed to predict how much the fish are eating, which can predict growth. And so this is kind of a lot to talk about in 5 minutes, so let's just kind of hone in on this temperature effect. So as expected, temperature had a net positive effect on recent growth, and that was direct. However, it had a negative effect on the abundance of prey in the environment and the negative effect on how much the fish were eating. However, the amount of appendicularians in the environment had a positive effect on how much they were eating and then how much they were growing. So this kind of seems like maybe a contrary finding, but let's look at the magnitude of these effects and compare them. So if we look at actually how large that growth effect is from temperature, it is so much more positive than the mediated effects from temperature on the feeding and diet. So what this means is though temperature can increase growth, the fish are likely able to eat enough to maintain it. So looking at all of our direct and indirect effects from that initial path diagram, distance from shore again kind of emerges as a negative driver of growth. So fish offshore aren't growing well, but interestingly, it's not mediated by environmental effects, which were included in this. And overall, other environmental conditions did not really contribute strongly to model prediction. So what does this all mean? I did a lot of work and a lot of modeling to discover that the direct effects are much more important for predicting larval growth than the indirect effects. And what this ultimately means is that actually larval English sole are doing okay under the range of conditions they're currently experiencing as long as they stay close to shore. So thank you to the Bill Wick Marine Fisheries Award, everybody who helped me, didn't help, it wasn't my project. We all had to go out in the winter in Oregon on boats, and it was rough. So thank you to everybody who helped facilitate collecting all of this data.
You've been trained. All right, everybody. How about one more big round of applause for all of our presenters today? And thank you all for being here for this event. As you can tell, and the soul of who we are is our graduate student researchers. And so I'm just really excited that they were able to share with you. In this time of uncertainty, we're also just incredibly grateful for our donors. It's just a time when it's just really nice that they're here to help us out and to help our students. And we couldn't do it without them. So I'm just really appreciative of them. So how about a round of applause for our donors as well? Thank you, everybody. So I also want to recognize some folks that you don't see standing up here that also make our student awards possible. In the financial office, we have Helen Britton, who works with all of these students all the time to help us navigate all of our funding. So I just wanted to recognize her because she's never seen. So thank you very much, Helen. And for this event, we had a lot of folks that have been helping with the food and setting up. So Shannon Reed, Marnie Zerbel, who was here doing our AV to the best of our ability, Katie Ergman and Miguel and Tammy O'Connor have all been here to make sure that this event was successful. So one more round of applause for all of them. All of our speakers did amazing. You did wonderful. Thank you so much. So we're a little bit ahead of schedule, but for those folks that are invited to come to dinner, so any speakers, their mentors, and any invited guests, we will have dinner. It'll be provided over in the same room where we had snacks. It probably is not quite ready yet, but I believe it's still sunny and beautiful outside, even though we can't see it. So dinner will officially be served at 5:30. I hope you stick around and join us. Again, thank you so much for being here, everyone. One more group photo, sorry. All our speakers, if you come up to the front, we'll get one more group photo.